Good afternoon, everybody. Welcome to another exciting CMBB seminar today. Um, this seminar is sponsored by Center for Molecular Biology and Biotechnology at Florida Atlantic University. CMBB is an exciting and growing program hosted by FAU and made of a variety of researchers and faculty members with a broad spectrum of research interest surrounding the biotechnology and the molecular biology. And it also composed of young and exciting students participate in this program. And if you want to know more about this program and about FAU, please visit us on our website. So now it's my great pleasure today to introduce Dr. Liu Lei from Harvard Medical School. So Dr. Liu did his medical training in Harbin Medical University in China. So in there, Dr. Liu obtained medical doctor degree, and he went on to do postgraduate studies at Shiga Medical School in Japan. And there he worked with a variety of faculties and then obtained his Ph.D. And from that point, his research focus has been on dementia, Alzheimer's disease, and the molecular biology mechanisms behind it. So today, Dr. Liu will give us an exciting seminar, introduce his recent work about the protein complex of secretase and their biological function and the meaning in Alzheimer's disease pathology. Dr. Liu obtained numerous awards and published high-profile papers in great journals. Currently, Dr. Liu is an instructor at Harvard Medical School Department of Neurobiology and also a research fellow at Brigham and Women's Hospital in Boston. So without further ado, let's welcome Dr. Liu.
Thank you for inviting me to this great seminar, and I'm really glad to give this presentation to talk about some of our work here in Harvard Medical School and the Brigham Women's Hospital. So I'm an instructor of neurology working Dr. Danny Selko lab. Dr. Selko is very famous for the amyloid hypothesis. And today we're going to talk about a cellular protease complex generates amyloid beta and its implication in AD. So the first I still want to introduce some of the background and how the study in the years came to this progress, because a lot of people might not be very familiar with Alzheimer's disease and how the molecular biology behind Alzheimer's disease. So I will keep this simple. And so this is some data from 2015 about demography of dementia. So maybe now the rate is higher, that we have one dementia in the worldwide every 3 seconds, or even less than that. So we see the increasing of the dementia patient across the worldwide, including the high-income country, middle-income country, and low-income countries. And specifically, the number of patients is increasing in Asia. So we are seeing kind of a pandemic of the dementia across the world, and we will see this very striking problem in the next coming decades. And this is another importance of the dementia research. So if we think in the total number of patients, mild cognitive impairment, which is the early form of dementia, and dementia only compose very few of the all the chronic disease patient. But if we see the growth, the MCI, the mild cognitive impairment, and dementia, that grow the fastest compared to some like a major cancer, like other psychiatric problem or chronic disease. So then these are a huge challenge in the coming decades, and how we can couple with this will be very important. And the next is some just general aspect of Alzheimer's disease. Alzheimer's disease is the most common form of dementia. It causes memory loss, difficulty performing daily tasks, changes in judgment, reasoning, behavior, and emotions. And nearly 60% of the case go undiagnosed. So then the early detection is very essential. Sorry. However, if we see the progression of the dementia, so here is the x is the time. There is when the people are cognitive normal, mild cognitive impairment and dementia. And here is the changing of the biomarkers in the cerebral spinal fluid, in the brain structure, memory, and the clinical functions. We could see the shift of these indicators of the disease through the time. But the time span is around 2 to 3 decades. So before the symptom onset, there are already 2 to 3 decades, the pathogenesis is ongoing. So how to manage a disease like this? So this is a very big challenge. And currently we have several efforts. Can we predict the disease onset earlier? So such as 10 years or 20 years before the disease onset. And this is about the diagnosis. The second is how we can tackle after they already get dementia. So there is already tremendous amount of neuronal loss and neuropathology, how to compensate the functions, and also how to have disease modifying therapies targeting the phase when the patient is undergoing here. But above all, no matter diagnosis or therapeutic, we have to understand the core of the disease. So then we came up to uh, actually a cascade which was studied for more than a century, is amyloid hypothesis. So here I put this timeline of since the identification of the first Alzheimer's disease patient by the Dr. Alois Alzheimer, and the identification of amyloid beta, and then the ongoing. So this is until like 2000, because actually the major findings from the genetic and the pathology was majorly conducted in the 1980s and in the 1990s. After the 2000, it's a century of molecular biology. We have more like genetics and something coming up, but I will focus on this kind of great like path towards a discovery of A beta. So the amyloid beta, or previously it's called A4 protein. Actually, the name is really just amyloid with beta sheet structure. It's called amyloid beta. A4 is amyloid with 4 kilodalton molecular weight. So this name is really just depicting what they are. It's not related with their genes, their anything. These are named, termed by their biochemical and by their biophysical characters. So if we see this is the report by the Dr. Alzheimer's, Alois Alzheimer's in 1911. This is the drawing actually of the observation under the microscope. You can see this is a round because this is how you see from the scope. And this brownish, yellowish stuff is amyloid plaque. And we also see there is neurofibrillary tangles, and there's a loss of cells So there's many like modern days observations actually is enriched in this one drawing. And the first report was actually presented by Dr. Alzheimer in 1906. But like other many important discovery, in the beginning, people just ignore it. Until 1911, people began to appreciate what's this really about. It's a very important histology challenge and which Dr. Alzheimer's discovered. And another is this kind of amyloid is not only in the cerebral cortex, it's also in the blood vessels. So from a review paper by Dr. Greenberg, who is very specialized in the cerebral amyloid angiopathy, this kind of disease. And so this is the A beta plaque in the cortex. This is the A beta aggregation decorating the blood vessel in the megansy in the brain. So, and this is something like eye my staining. This is I stained the patient brain like almost like 10 years ago. This is around 2 years ago, both by the chromogenic histology and the fluorescence histology. Like there's something like you cannot escape from this is when you see a AD patient's brain like this, the A beta plaque is occupying the whole cortex. This is so striking. So when you like in the microscope room in the dark, and you can see the whole. So for example, this is a temporal lobe. It's glowing. This is glowing with amyloid beta labeling. So then after 1906, this very nice immunohistochemical data was presented. After almost 80 years, there's finally some like next step progress. So I will show in the next slide. So this is 1984 of two papers published in the BBRC by Dr. George Glenner. And this paper was the first time which he extract the blood vessels from the AD patient's brain and isolate the fibril and identified the amino acid sequence of the amyloid. So that is very interesting because Dr. George Glenner, long time he speculate that the cerebral amyloid angiopathy, which I showed in the blood vessels, and the amyloid plaque in the cortex should be the same thing. But at that time, 1984, he didn't know that because it's so difficult to purify the amyloid plaque from the human brain at that time. So then he chose a relatively pure or clean samples, which is blood vessels, because blood vessels is majorly is smooth muscles. There is connective tissues. You can just digest it with collagenase, and you can release these fibrils. So just 1 year later, actually, after 1984, he found both in the AD patient brain and the Down syndrome brain, these kind of fibril. Uh, then in German, there is another group found like in the amyloid plaque in the brain cortex, also compose the same amyloid protein. So here he called it amyloid beta, and in the other paper they call it amyloid A4. In the end, they are the same. They are the same protein, same sequence. So then after 1985, it's a kind of exploding of the amyloid field. So then there's a two paper in 1987. They cloned the gene of the containing the amyloid beta sequence. Amyloid beta protein is only 4 kilodalton. However, what they discovered of this precursor of amyloid protein is very big. It's more than 100 kilodalton. It have many truncation form. The longest is a 771 amino acids. So then after the APP is cloned, And then there is the first APP mutation, which is London mutation, was identified in 1991. And the amyloid hypothesis then began to be built up in 1992. So, and again, like in 1995, like there is a presenilin, which was the catalytic component of the enzyme, which generated beta was cloned and founded in University of Toronto. So all these history is very like telling us that once the A beta was identified, it's almost a field is born, a field of biochemical characterization of amyloid and its precursor and its generation aggregation has just started. So then we go go to see the amyloid beta or A4 protein precursor is APP. So it's a very big protein. It has many motifs. It have a signal peptide, HBD binding domain, copper binding domain, and a lot of conserved like function structure domain. And A beta is just happen to be in the transmembrane part, and also there is an intracellular domain. So for a long time, we don't know that how the A beta is generated from. So until we found like A beta is a release from this amyloid precursor protein by the protease proteolytic events. So the proteolytic event is termed by hydrolysis of the peptide bond inside the protein. So we have different enzyme, as I showed here, beta secretase, alpha secretase, gamma secretase, eta secretase. So I color by different scissors. You can see they cut different part of the APP. Only the beta secretase and the gamma secretase, these two will release the A beta, amyloid beta. So amyloid beta is termed by its extremely hydrophobic nature. So it can form in the beta sheet structure. So then it can deposit outside of the cells extracellularly. And the gamma secretase was identified. The core of the gamma secretase is a presenilin, which is a catalytic component, was identified in 1995. Beta secretase was later, 1999. So since then, like people began like a very extensive exploration of how these hydrolytic, like hydrolysis events happens. So this is a very complicated process. So here I blow the A beta sequence into bigger. So here's from the A beta membrane 1 and to the A beta membrane 53. There's no A beta 53, I just including all the sequence here after the transmembrane domain. So the first cleavage of the A beta, it cut around the here is like it generate either is 49 or 48. And then it's followed a tripeptide trimming pattern, two different pathway, A beta 49, 46, 43, 40, and 37, or A beta 48, 45, 42, and from the 42, there's multiple options. You go to 39, 38, and 37. So why is the cleavage pattern like that? So we began to understand this. So this is a helical structure in the as the transmembrane domain. So the helical structure cannot be hydrolyzed. It has to be stretched and linearized. So every time the linearization of the helicity is a one round of the helix. So as we know, the perfect alpha helix will have averaging 3.6 amino acids. So this is why we are having like one step of the linearization is a 3 residue. So we have the 3 residue trimming event at the same time. But if we go to see in the brain, in the CSF, in the plasma of a patient, or even a normal people, because the APP, this is not only in the brain, it's in almost every single cell of our body. So we are generating A beta every second, every like every moment. We might generate as much as like 5 milligram A beta per day. And at the same time, if we think this is a perfect enzymatic reaction, we want all the product into the end product, which is A beta 37 or A beta 38 or A beta 39. But unfortunately, because APP have a specific character that they have this GXXXG motif, which is loosen itself from the enzyme, which causing there's a consistent level of A beta 40, A beta 42, and A beta 43, and like longer than A beta 37 and 38. So these A beta will have a longer C terminus, which including C terms like here. If we see the VVIA residue and until the T, they're extremely hydrophobic. So these are the problem. So which means even we have no mutation of APP in our body, the A beta generation and A beta aggregation seem to be uninevitable. So this is another feature of APP, like we recently published a paper in the GBC. If you guys are interested, you can look for it. But today we're more looking at how the beta secretase and the gamma secretase working together to generate these A beta. So normally when we think like enzyme, especially the hydrolysis, you need water. So how could a hydrolysis happen in the transmembrane where there is extremely hydrophobic environment? So this is the first time the it's called regulated intramembrane proteolysis was introduced. It was SREBP1, a membrane bound transcription factor. It actually was a very, very interesting phenomenon that on the membrane there is a sterol. Once the sterol is depleted, they will trigger a cleavage event by two enzymes. First by S1P and second by S2P. So S2P is catalyzed intramembrane and then release a motif, enter the nucleus, which as a transcription regulation of the lipid and fatty acid metabolism. So this is a perfect case of a self-evident like a loop of modulation from the memory to the nucleus to the transcription. So these very important signaling was identified by Dr. Goldstein and Michael S. Braun, Dr. Braun. So these two very famous in the lipid metabolism. And so then they introduced this regulated intramembrane proteolysis, which is similar to the gamma secretase. And then we came to a point that if we have the gamma secretase continuously generate A beta and A beta is bad in some way, like can we stop it? Like can we promote it or can we modulate it? So these things are the questions we have for a long time. So then like here is a kind of a schematic is how A beta is generated by these regulated intramembrane proteolysis. We said we have a type 1 membrane substrate, which is such as APP. And we have a sheddase, which is a term as beta secretase, which cut these type 1 membrane protein, transmembrane protein around the ectodomain. It's still like either it's extracellular or it's in the luminal. And then you have the gamma secretase to further cleave this kind of transmembrane part and into a secreted form as A beta or its intracellular part as intracellular domain. So whether they go to the nucleus is still in question. For Notch, another very important signaling for neurogenesis is sure. But for APP, we're not so sure whether this is a transcription factor. So this is the model for a long time. We have saw like first there is a substrate in the membrane, they met the sheddase, and then there's a cleavage. And then this kind of small part of the APP is floating on the membrane until it met the gamma secretase. and to mediate the cleavage. So I called it model one because this was a previous model before we start to hypothesize alternative. However, I will go back to this slide later. So we have different thinking of this model. The reason is we want to find the biological rationales behind a multi-secretase, like several enzymes work together. The reason first, a beta release is a synaptic activity dependent. And so this was very nicely shown by Dr. Cerrito and Dr. David Hausman in WashU 2005. So when they have electronic stimulation of a brain slice, they found that the basal A beta secretion is increased after the electronic stimulation, which is kind of boosting the synaptic activity, which we can see by the recorded EEG activity. So another thing we know that synaptic activity is pretty fast. Synaptic activity is in the scale of millisecond. So how come a millisecond event and A beta can follow that? If the APP is floating on the membrane for the first enzyme to cleave and for the second enzyme to follow, it's not that efficient process. The second thing is we found in our lab, it's by Ellen in our lab, we found that APP actually is the full-length protein interact with the mature gamma secretase. If the first model was correct, the full-length APP have to be cleaved by the sheddase to meet the second enzyme. So how come the full-length APP could meet with the associate with the mature gamma secretase? And then, so the Alan in our lab, he continued to explore and he found the physical and function interaction actually between the alpha and the gamma secretase. So alpha secretase is similar to the beta secretase. It just generates different APP product. So then with these indications and the previous reports, and then I began to think whether the sheddase or like here, for example, is the beta secretase and the gamma secretase, they work together and with the full-length APP to efficiently generate a beta, ASCD, and the soluble APP at the same time. So then we began to examine this. So I will show this kind of example of how to examine first protein-protein interaction. Second is a functional enzyme, right? Whether an enzyme is a function. So then, however, when we go to the real issue here, if we see the structure of the gamma secretase and the beta secretase. So gamma secretase itself is a gigantic complex itself, right? It have 4 different components. Have a presenilin, nicastrin, a pH, and a PEN2. Totally, they have 20 transmembrane domain. And the beta secretase is relatively simple. However, how to study these complex is very difficult. And can we study them natively and relatively natively under the native condition is a very tricky issue. So then we have a checklist of things to do, how to examine the existence of the beta secretase and the gamma secretase complex. So all these data I present today, most of them you can find in the paper we published in 2019 in the Journal of Cell Biology. So in case you want to see more details. So there is a several questions we need to ask. The first, we want to show like, do they physically associated? And the second is what kind of complex they form. The third is how stable is this complex. The fourth is this complex functional? The function here is enzymatic because they are enzymatic complex. We need to test their behaviors from the enzymological aspects. The next is what's the mechanism or like necessity of it. The next is, do we have any pharmaceutical intervention? And the last is this enzyme complex universal mechanism for intramembrane signaling? So then we answer these questions one by one. The first is, does they associate with each other? Because the gamma secretase is a very big complex itself, we need to test whether the beta secretase interact with each of the components. So then with the immunoprecipitation, we very carefully did this with the three different negative controls. They're all transmembrane proteins and also with the beta secretase. We could see the beta secretase could put, antibody could pull down the gamma secretase component. The second thing is when we fraction the wild type mouse brain under like detergent, this 1% capsule is a very mild detergent, could maintain the gamma secretase integrity. We found that the beta secretase and the gamma secretase, they co-migrate on the two-dimension gel, which shows a quite big molecular weight. It's about 480 kilodalton. And if we use a harsher condition, such as another detergent, DDM, we could see that this harsher detergent will break down the base one and these components. So they are no more aligned. So if you see here, these complexes are aligned together on the gel. Here, they are no more aligned with each other. So this is the first evidence that gamma secretase complex associated with the beta secretase. The next is like we want to study these complex in the native form. So then we choose a size-exclusive chromatogram using the 1% capsule. And then we, after refraction the mouse brain, and we found like the beta secretase, gamma secretase, there is a all migration overlap in the very high molecular weight fractions. So this is bigger than 5 megadalton or 5,000 kilodalton. However, the interesting point is only the 10% of the base one or beta secretase co-migrate with the gamma secretase. So it's representing that the complex is only a very small portion of the beta secretase. So there is also free beta secretase allowed. And then like also using the immunoprecipitation, we pull down from the size-exclusive chromatogram fractions, we still found like the base one antibody could pull down the gamma secretase components in the high molecular weight fractions. And another thing is after we using the synthetic beta secretase substrate, we using the gamma secretase antibody to pull down from the brain, we found this pull down has the beta secretase activity. So which means this complex is enzymatic active. And the next is using the cytochemistry to localize the beta secretase and the gamma secretase. So we stained for the presenilin 1 and the beta secretase base 1 in the HEC2 necessary cells. So this is just the double labeling of in the conventional confocal. And after we use the super resolution, the nanoscope using STAT, after the laser depletion, we could find that very fine like dot of the structures in the cells have these two antibody double labeled. Like moreover, we're using a proximity ligation assay, which the two antibody can polymerize if they're within 40 nanometer proximity. So we could see that compared to the control condition like PS1 and the transferrin receptor, the PS1 and beta secretase have huge like a positive signal, which indicating they're at least presenilin and the beta secretase, they're at least within 40 nanometers. And in here, this is the resolution is even higher. So we could see they're within like 10 nanometers around that. So then from the immunocytochemistry, we also came to the same conclusion that gamma secretase components associated with the beta secretase. So the next is we go back to the model. So what will be the next is we want to show that this complex is functional. So getting a functional complex from cells or from brain endogenously is always challenging. So then we just tried see whether we get some luck. So we get cells. So this is hectonatory cells with overexpression of the APP. So this APP is the substrate of the beta and the gamma secretase to generate A beta. So we fraction this on the sex exclusive chromatogram. We found a similar pattern as we see in the mouse brain. The 2D dimension gel also the similar. And then we use these fractions. We use these fractions because there is a beta secretase, there is a gamma secretase, there's also APP. So then how about we simply just incubate? So we incubate this fraction 37 degrees for overnight. We found there is a beta, both a beta 40 and a beta 42 generated from these fractions. And if we don't incubate them or we put them 4 degree, we don't see these signals. These are measured by ELISA. We call them de novo A beta generation from these sex exclusive chromatogram fractions, and which indicating that the enzymatic complex, if there are enzymatic active, they can process the APP into A beta. And the second is we have the A beta 42 to 40 ratio, as I mentioned before. A beta, there's a panel of A beta generated by the gamma secretase, and there is relative ratio. So we found that the most fraction generated A beta will have a 42 to 40 ratio below 0.2, which is kind of physiological ratio. But still, like from this point, we know there's A beta generated from these fractions. Where does these A beta came from? Is it from the APP full length by 2 enzyme, or is it from the APP truncated by only gamma secretase? So as we see here, we go back to this slide. If the beta secretase already cleaved the APP, we will have this part. If this part is called a C-terminal fragment, if only the C-terminal fragment is cleaved by the gamma secretase, we still get A beta. So then we need to further explore. So the next is we want to show whether this enzymatic activity can be blocked or be modulated. So then we use two types of compound. One type of compound is called a gamma secretase inhibitor. It inhibits the gamma secretase activity. But when we throw this compound in the fractions, we didn't see any blocking effect. So we're kind of puzzled. However, when we use the other type of compound, which is a gamma secretase modulator, the modulator will increase the short A beta production and decreasing the long A beta production. When we throw this compound in, we do see very good response. From these fractions, we see the decreasing of 40, decreasing of 42, and an increase of 37 and decrease of 42. So then why the gamma secretase inhibitor didn't work? So then we hypothesize that when there is already the gamma secretase, beta secretase, APP, the substrate is already docking into the gamma secretase. When we are using the transition state analog, which is a substrate mimic, it is no more able to enter the gamma secretase substrate pocket. So because this compound is not that potent, so later we try much potent, which is now under the clinical development. There's two compounds we get from Dr. Rudy Tenzi and Steve Wagner. And we try these two compounds in these fractions. We could see the 40, 42 decrease and a 37 increase by these modulators so dramatically, only by 50 nanomolar. So again, these experiments show that the enzymatic, these complex is enzymatic active. So then we go to the human brain. So whether we can see the same thing from the human brain. So this is the Western blot from the human brain fractions. We see the gamma secretase component, beta secretase component. Another thing is we can even produce de novo A beta from the human brain fractions by the sex-exclusive chromatogram, as shown here. So this is a very like a fresh brain I indicating here because the postmortem time is very short. We directly process with the membrane fraction, and it's generate a lot. Here you could see generate 100 picogram per mil. And then when we test whether when the human brains are frozen, we test on this modulator. It's not so well response by the 40 reduction, but it still responds as we see by the increase of the short A beta production from the human brain. So now we can conclude that from the cells, from human brain, we could observe this active enzyme, and the enzyme could be complex. So then the next question is, what is a substrate from this high molecular complex A beta production? So we have two hypotheses. It's from the CTF, which is already processed by the beta secretase, or another option is that they are processed by the full-length APP. To do that, we pre-treat the cells, APP cells, APP HEC293 cells, with a beta secretase inhibitor. So with that treatment, we will not have APP CTF beta anymore. So then these cells already deplete all the APP CTF. They only have APP full length. So then we take these cells, fraction them, and we compare the control group. We could see that the novel production of the A beta boosted by at least 4 to 5-fold. So then we are more confident that this A beta generated from this de novo production is actually from the full-length APP, because in this treatment group, there's no APP CTF, there's only full-length APP. As we show here, after the treatment in this fraction, there's very much enhanced of the mature APP full-length protein. The mature APP is the only APP can be processed by the beta secretase, followed by the gamma secretase. So this is a very solid evidence that we have like the secretase complex, and they are processed from the holo APP. But still, like we're dealing with a mixed system. We have the fractions are now pure. They have a lot of other components. So then we began to think whether we can further isolate these beta gamma complex. And then we have to expand our like a scale. We have to increase the scale. So we change from the analytical column to a preparative column and do the same thing. We culture a cell. Actually, it's a lot of cell. It's a 15-centimeter dish. We have 15 dish of them. We prepare the capsule lysate, pass through the column, and we pull these fraction together. And here we're using 4 different antibody to isolate the potential beta gamma complex. They can recognize presenilin, gamma secretase, beta secretase, and also APP, and full-length APP. In this case, we're using an N-terminal APP antibody. So this is how the fraction goes and how the IP goes, show the successful IP and a co-IP of these proteins. And then the next is we found when we take these IP, so APP, IP, nicotine, the gamma secretase IP and beta secretase IP, we could find A beta de novo production and A beta 42 de novo production, and also these immunoacylated complex are response to both 2 type of the gamma secretase modulator. So up to here, we can show that its APP full length can be processed by the beta and the gamma complex and into A beta, and these enzymatic reaction can be modulated by the gamma secretase modulator. And then like we also did some like explorer, we want to show whether the transmembrane domain of the beta secretase is critical, which exchange to another transmembrane protease. However, unfortunately, we didn't find any positive findings. So the transmembrane domain of base 1 is now specific for this beta and gamma complex formation. So the next is we want to ask, can we modulate this complex? Can we modulate the formation of this complex? So through screening a lot of compounds, I found a natural compound, ruburic acid, is a very unique natural compound. It's a pentacyclic, but the A ring is a cycle A ring. So this compound could potentially destabilize the beta gamma complex. So the first we test, we treat the HEX cells, we found a beta reduction dose dependently, and then we found like, okay, so whether this compound have the potentials. So then we test something on the sex exclusive chromatogram. What we found is that once we treat the HEX cell with this ruburic acid, we found the in the hymercoate, all the gamma secreted components began to decrease. So simultaneously, the A beta production from these fractions also decreased. So which is indicating that there is a depletion effect by the ruburic acid targeting the beta gamma complex. And moreover, we found like they have a more potent like potency towards A beta 42. So as we show here, when we use the ruburic acid treating the A beta, treating the iPS-derived human neurons, we found the A beta 42 to 40 ratio decrease and 38 to 40 ratio increase. So the ruburic acid actually is a double function reagent. It both can destabilize the beta gamma complex to reduce the total A beta production. Second is it also can modulate the gamma secretase to promote the shorter A beta production. And to know that the shorter A beta species are non-toxic. They have less potency to aggregate into the oligomeric A beta and the fibrillar A beta. So getting the shorter and the less A beta is always a good sign of a therapeutic target. So up to here, like I think we use all different level of the experimental design to conclude existence of the beta gamma complex. And let's go back to our first part of our biological rationale of the beta gamma complex. Why there should be a beta gamma complex? Because we need an efficient generation of the A beta. So we want to study whether the synaptic or just the general activity of the cells, like is dependent A beta release correlated with the complex depletion. So we treat these iPS-derived human neurons with a potassium chloride to induce a polarization. We could see the A beta secretion jumped after 1 hour treatment, 30%, which is exactly the same when the John Cerrito and Dr. David Hausman found in 2005, which is a 30% activity-dependent A beta generation, more A beta generation. So then we just fraction the cells, and what do we find is that there's a little bit depletion of the like the complex. If we see after the 1 hour depolarization of the iPS-derived human neurons, the APP holoprotein, they decrease dramatically in the hymercoate. But after 12-hour treatment, we could see the rebounds of the APP holoprotein in the hymercoate fractions. So this is not a very yet optimal experimental design because the iPS-derived neurons are not fire very well as a neuron. So they are not typically a real neurons. So we are trying to make a real neurons and to do the same experiment to see whether they still stand there, that the beta gamma complex has a direct connection to the activity-dependent A beta generation. And so this is the summary of our findings. So we found that the gamma secretase and beta secretase and APP can form a complex together. When we use the base inhibitor, there's more of the de novo beta generation because there's more APP holoprotein. When we use the gamma secretase inhibitor, there's nothing changed because there's a complex already there, the substrate is already docking to the enzyme. When we're using the ruburic acid, we are seeing the de novo A beta decreasing because of the complex is destabilized. When we use the gamma secretase modulator, we could see the de novo increase of shorter species, decrease of the longer species. So from in the beginning, we have the checklist. So now we only have the last question is remained. We have answered all these questions accordingly using different technique, immunoprecipitation, exclusive chromatogram, and enzymatic assay, ELISA detections. And the last will be, is this enzymatic complex is a universal mechanism for intramembrane signaling. So this is a very large question. We will test a lot of different substrate of gamma secretase. Gamma secretase has more than 100 substrate, and also the beta secretase has more than 50. Another is this shared by other proteases at the same time. As I first introduced of the regulated intramembrane proteolysis, the SREBP1, the very critical lipid metabolism transcription factor, is processed by the SET1 protease and SET2 protease. Does these two proteases also share the similar mechanism? That will be very interesting to know. And so the next is that today I only introduce a very tiny portion of what I'm doing and I was doing. So this is a workflow of my a daily job. As I'm a beta person and I'm pre-selling person, I have a roadmap of different works focused on the life of a beta. I've focused on the pre-production of the A beta is today's contents. I also focus on the production of A beta, which is the enzymatic characterization of gamma secretase. Well, I also focus on the post-production of A beta. I do like a biomarker from the plasma, CSF to measure how much A beta monomer, oligomers, and there's also biological consequence as how does these A beta in the plasma, CSF, brain correlated. For example, the most direct consequence is the hyperphosphorylation tau and the neurofibrillar tangle, and also other like inflammatory response and how these symptoms related with A beta. So everything actually go back to the A beta because as I shown in the first couple of slide, my stainings and other people's stainings, when you look at a real AD patient's brain, A beta is just something you cannot escape from. And maybe you want to escape from, but it's just so hard. You cannot ignore it. We have to get an answer from it. And that showed that A beta is a kind of, they will aggregate anyway because of the APP's character. So we also need to find a way to spin off, to pull off how it's aggregate and prevent the symptom to be onset and so on. So my work covered both therapeutic and diagnostic. So mostly nowadays we do transformative research. And in the end, I want to thank to the people in my lab, my mentors, and Dr. Dan Selko. Like if without him, none of the research is possible. And also our funding resource, I'm funding by NIA and also MEDRC. So here's my email address. If you have any further questions, please feel free to shoot me a email, and I'm really happy to answer any questions. Yeah, thank you for the attention, and maybe we can go to the discussion.
Thank you very much for Dr. Liu's work. Given the complexity of your discovery, that the existence of the complex alpha gamma complex and beta gamma complex, and then given the complexity of the A beta cleavage, what do you think about the relatively simple amyloid hypothesis that have been used to develop pharmaceutical treatment and have been experienced a lot of failures during the past 2 decades, right? So, as you said, from 1992, the amyloid cascade hypothesis has been established. And obviously, it has been established on very strong evidence from familial assembly study, from enzymatic studies, all sorts of studies. But however, building on this seemingly very strong hypothesis, most of the pharmaceutical drugs developed or the clinical trials hasn't been successful so far. So what do you think, based on your discovery and based on the pharmaceutical industry's result, that what we should treat and understand or improve or modify the current hypothesis in terms of the disease pathogenesis?
Yeah, so this is a very great question, a very tough question, and I think I want to answer this at the same time with the question in the Q&A section, the first one. So like the question is giving the large number of beta and gamma secretase substrate. Yeah, so um, beta secretase and gamma secretase has many, many substrate, and a lot of them is very critical. And we could see the consequence of inhibiting them, like both we have the segmastase, very big fail from the Lilly, and we also have all the beta secretase inhibitors failures like a couple of years ago. Actually, we do conduct some post-mortem study of these inhibitors. So the first thing is we have a concept of the A beta hypothesis is as long as we stop A beta production, it's good. However, that is not doable because when you have the enzymatic activity like every second, every like all your cells, it's almost like impossible to stop the A beta production. So this is the we should like think twice about any inhibitor idea or like inhibit the gamma secretase activity or beta secretase activity because from the beta secretase inhibitor failures we learned it's impossible to design a substrate specific inhibitor. Gamma secretase inhibitor is same. And so this is why we focus on the modulation of the gamma secretase because the only problem of A beta, it's not its production, it's aggregation. A beta aggregate only when you have these like VVIA, these very hydrophobic residues. So when you modulate the enzyme, you will reduce these VVIA residues. So you get A beta 37, which we found they never aggregate. I only found like a one plaque, two plaque in the whole brain sections, human brain section labeled by these antibodies. When you have these shorter species, you definitely find. And another thing is we don't understand how does this A beta aggregation connect to the toxicity. So that is a very like long debate. How does A beta? So we more and more came to a hypothesis that A beta, the existence of A beta is not presenting as a toxic. So it's rather than it's a progressive, the aggregation. If the existence of A beta aggregates is toxic, the pathogenesis won't last for 3 decades. Like it's almost like you get a problem within half a year or 10 months instead of like 30 years. So to understand that is also like very, very important. So what I'm thinking is rather from the small molecular like way, because most of my work on the small molecule. Antibody has this advantage. It's like antibody biology is beautiful, but we have to encounter the problem is only less than 0.1% of the antibody can pass the BBB. So if we think of the small molecule, I would suggest a compound don't inhibit beta secretase or gamma secretase rather than modulate. So some as we term as GSM. So that's one classic. The other is the one like we identify this rubiuric acid. It don't inhibit the activity, but it separates the complex. So then the signaling of the cell signaling is not interrupted. But as I showed, this is natural compound. It's almost like impossible to modify. So it's a tough road to search for better small molecule. So that is for sure. So Chi, you are unmuted. You are mute. Yeah.
So get used to it.
Yeah.
So the second question in the Q&A board, is there any evidence that the formation of your complex or activity of your complex is affected by hypoxia or oxidative stress? And in my personal addition is that, as you pointed out, these events of cleavage happen on the membranes, right? So the
Yes.
cell membrane is also affected by a lot of cellular processes of stress, etc. Just like when the audience asked. So, what your comments or your opinion on this?
Yeah, so that is good question. Like we didn't test the hypoxia or like oxidative stress uh, because we mostly focus on how the enzymatic reaction happens. Because when you have a hypoxia or oxidative stress, you have a completely whole cellular pathway be open, like a different cell signaling. What we did is we try to deplete the cholesterol. We try to disrupt the lipid raft. We treat the cell with a cholesterol oxidase. So that oxidase only oxidated the three hydroxy group of the cholesterol. So it no more form the lipid raft with the sphingomyelin. So using that, we could clearly see the complex is be actually liberated. The complex be liberated, and because the complex under like a stable station, like under the stable status, they're not active. So there is like a sterol, fatty acid, and lipid separate them, bounce away of them each other. Once you deplete them, they are ready to generate a beta anytime. So, however, like from this is very artificial experiment. So we have to go back to see if the any hypoxia can relate it. But one thing I can tell, hypoxia will induce a beta production. That is through the HIF1 signal. HIF1 signal will induce the beta secretase expression. So then you have more beta secretase, you have more A beta. That is tested by many like animal models. You do an MCO model, you have more A beta.
And then the third question is that the heavy metals, in terms of its influence or effect on the cleavage, on the complex. Can you comment on that?
Yeah, so like heavy metals is hard to study as always, but what we know about is one component of gamma secretase is APH1. APH1 have his six histidine in the transmembrane domain. So a long time we believe it's kind of like a zinc cage or something like that, and but we don't have evidence for that. However, the heavy metal like it's really not my field. I have never tested that.
Okay, the fourth question. So again, this person, Guan Lopez, I think, is actually also a research on the gamma secretase cleavage, etc. So his question centered on, does the alpha secretase also form complex? And I think you touched on that briefly. So if you can give a little bit more details.
Yeah. So the alpha and the gamma secretase also associated. So either it's Allen's work in 2015 or like my data can very strongly approve that. And I tried some like de novo experiment to generate NICD, which because the like complexity of the nutrient biology is not that details. So I haven't have any success yet, but I would predict that the alpha secretase and gamma secretase form a complex, and there's more abundant because alpha secretase has more substrate than beta secretase. That is one. The second is if we take APP as a example, like APP, 95% of APP is cleaved by alpha secretase than the beta secretase. So you will see more of that. It's much easier to identify.
And then last question is actually also a question I think much relevant to to the sporadic Alzheimer's disease. The ApoE4, right, is the highest genetic risk factor for sporadic Alzheimer's disease, which is the most common form. So can you comment on that? How the ApoE4, people have ApoE4 is being affected, and then how you interpret that from the complex point of view?
Yeah, so like we didn't look at the ApoE4 and ApoE3 brains because from the brain tissue, it's almost impossible to do any like a quantification of these complex neuronal loss and all things extra. What we have done is we test the A beta secretion from the ApoE33, 22, and the 44 like IPS neurons. So, but these are not like naturally from patient. They are like corrected, CRISPR, like mutated. We do find the A beta production is a very much difference. So from what I understand is Dr. Tom Sudov like published about like ApoEs related with A beta production. I'm not sure our data is really supporting that, but I think it's something we should explore. At least we see the A beta production is different. But whether the difference is came from the beta gamma complex or any other, it's release intracellular A beta, that is on like we don't know it yet.
So I think this concludes Dr. Liu's talk for today. And as I said in the beginning, if anyone's still interested in in-depth discussion with Dr. Liu, you can stay and I will turn on your video and audio so you can participate in a one-on-one in-person discussion. All right, thank you everybody for attending this talk. Great talk, thank you.
Thank you everybody. Thank you for attention.